go to our cell unit. And so what we're going to start off with is sections 4.2, 4.3, and 4.7, which just outline kind of the basics. And a lot of these things are things that we're going to remember. We won't get into too much detail with specific organelles. This is just the really basics of why we study cells. And really quick, one of the first things that we're going to be learning about is differentiating prokaryotic cells, like this one over here. And this is actually bacteria, or a bacterium, and eukaryotic cells. Right here, and this is actually an animal cell. And these are some really cool or nice looking microscope images that will kind of give us a visual of what exactly we're talking about. So why exactly talk about cells? Well, cells are the building blocks of life. All life starts from these cellular building blocks. You might think, what about viruses? Viruses, not alive. We'll talk about that later in this unit, but viruses not alive. That can be kind of why we say they aren't made from cells. But cells are the building blocks of life. They're the smallest part of life that's non-divisible. So we can't really break cells down into anything smaller and still retain the same identity, the same function. We can break down like a liver or a kidney smaller into tissues and then finally into cells, but we can't break cells down into any finer parts that still carry the same function, that still go through homeostasis or maintain homeostasis. When we talk about cells, like we just looked at, we like to divide them into two groups, into prokaryotes and into eukaryotes. And there's some differences between them, but it's one of the main important things to remember is what's similar between all cellular life. All cellular life has a plasma membrane, so that kind of wavy sac that surrounds the cell, holds it in, kind of like the skin of a balloon. All cells have cytosol, which is that jelly-like substance, jelly fluid, that's on the inside of the cells, that all of the things on the inside of the cell is floating around him. All cells also have ribosomes, or protein factories, that make proteins, little small studded looking structures. And all life has chromosomes, or DNA storage units. Now, even though prokaryotic cells have their DNA just floating around in the cytoplasm in things called plasmids, or plasmoids, and these are for prokaryotes, they're still considered to be chromosomes. You might say, what about the mitochondria? Plants everything needs a mitochondria for cellular respiration to generate ATP. You know what? Prokaryotes don't. All the enzymes and the things that would be contained in the mitochondria just floating around free in the cytosol of a prokaryote. Same thing with like the chloroplasts. Some bacteria, cyanobacteria, photosynthesize. All of those chlorophyll-like pigments and enzymes necessary for photosynthesis are just floating around free in the cytoplasm without specific structures. So even mitochondria and chloroplasts are absent from prokaryotes. So prokaryotes are the oldest and the simplest cells. They came about 3.5 billion years ago, only about 1 billion years after the Earth formed, which was 4.567 billion years ago. They're simple and small. They're about 0.1 to 1 micrometer at their smallest, and they can get anywhere on average from 3 micrometers up to 5 micrometers. They don't have any organelles or a nucleus. Important. Absent organelles or a nucleus. The genetic material, DNA, floats around in the cytosol in circular structures called plasmids or plasmoids, kind of like this. They're floating around in there. Now, looking at this picture, you can see some of the interesting characteristics of prokaryotes we'll talk about quite a bit later in the year. They have a cell wall, much like plants. The outer layer, the capsule, kind of protects the prokaryote and helps them stick on to different organisms, and then they have a double-layered cell wall and a plasma membrane. A lot of them use flagella to get around, and they have these projections out of their cell walls called pili to help them hold on to things. You can see that the nucleoid region, well, it would be the nucleus, but isn't it the big group of DNA or the chromosome for a prokaryote? Alright, eukaryotes are larger than prokaryotes. They're between 10 micrometers and 100 micrometers. And they can be multicellular. Prokaryotes, by definition, are not multicellular. Here's a good picture of a eukaryote. You can see it has a nucleus. This is where the DNA is found. And it also has all of these little things. They're organelles. They're organelles that are basically the parts that do specific jobs for the cell. So they have certain functions. Kind of like if you have a different job within a city, all the jobs help to keep the prosperity and the welfare of the city up, but everyone has a specific job to regulate the city, and just like a city, all these organelles have a special job to keep the cell alive and happy. Now, in the next couple days, we'll be going through a various amount of cell organelles in eukaryotes. Much of this section is on eukaryotes, and this is a good picture to distill all the ones we'll be learning for both plants and animal cells. The picture, the double page in your book in section 4.2 and 4.3 includes all of them that we're learning about, or that you'll be expected to know, and this diagram does a good job in distilling that information as well. So, why are cells so small? The human body has between 10 and 100 trillion cells. Why are they so small? Well, let's just think, what if they are large? If they're large and one cell died, that would be no good. Imagine if cells were the size of baseballs and one cell in your arm died. That's a huge chunk of your arm you just lost. So they need to be small to be able to reproduce and replenish and die. But also, cells need to maximize the resources they absorb and the waste they eliminate. And as we'll talk about later in this section, resources and waste go in and out of the cell through the plasma membrane. It's kind of like the gate. So, all the waste and all the resources, they have to go back and forth across the plasma membrane. If a cell is really big, so it has a big volume, it needs a lot of resources. A lot of things have to go to and fro across that plasma membrane to service the cell and to be disposed of, excreted. If a cell has large surface area, then it can transport a lot of resources in and out. So we're actually going to do a lab on this on Friday, but I won't ruin the surprise, but I want you to think about, why do cells need to be so small? Your book goes in a little more detail, but we're really going to explore it on Friday. But just a little hint, if you see root hairs, really small and thin, they're responsible for nutrient uptake. And in the human body, the alveoli is really puffy, round, with thin walls, and used for gas exchange. So maybe those two examples will help you think of why cells need to be so small. All right. So one of the things we're going to talk about in the eukaryotic cell is the nucleus, and it's a very big and easy to see in those pictures. It's a big, round, darkly stained circle that contains genetic information, the DNA, and blueprints, instructions for life. It's inside a membrane, and not just any membrane, a double membrane. So it has two phospholipid bilayers. Together it's called the nuclear envelope, made of phospholipids. We'll come back to that later. We'll do an intensive study on the phospholipid membrane. But suffice to say, the nucleus is enclosed by a double phospholipid membrane. And it has these little holes in it right here, called nuclear pores, created by proteins that bridge the double membrane, and that's called the nuclear pore complex. And it allows materials such as RNAs and proteins to go back and forth. And inside of the nucleus, we have the genetic information, the DNA, that is stored in chromatin. And in this picture, it's in a chromosome, because whenever the DNA is condensed in this X-like or rod-like structure, it's called a chromosome. But whenever the cell's not dividing, it's kind of in these strings of DNA called chromatin. Alright, so double phospholipid membrane with nuclear pores let things in and out. There's also ribosomes that are stuck on the nuclear envelope, and chromosomes / chromatin, when it's not dividing, store the genetic information. Inside the nucleus, there's a specially dark region called the nucleolus, and that is where ribosomes are made. And ribosomes are made out of rRNA. Easy to remember, R, ribosome. And in the nucleolus is where rRNA is transcribed and translated into ribosomes. So that's why it appears so dark, is because it's the site of lots of protein synthesis. So some of those ribosomes that get made in the nucleus become bound to the nuclear membrane. And some also get bound to the endoplasmic reticulum. We call these bound ribosomes. And these bound ribosomes produce things that are usually either used in the nucleus, like enzymes or different cofactors, or they produce things that are going to be secreted by the cells. So bound ribosomes produce a lot of enzymes, or things used in nucleus, or excreted material. So for example, a stomach cell like we have here would have a lot of bound ribosomes, especially to the smooth and rough endoplasmic reticulum, because it has to secrete gastric juices. In the other case, some other ribosomes are just floating around in the cytoplasm, and they're called free ribosomes. And what they generally do is make protein that's used in the cell cytosol. So muscle fibers would have a lot of unbound or free ribosomes, produce a lot of protein to build those muscle cells up. Cells that need more protein have more ribosomes. Lastly, all around our cell, there's something called the extracellular matrix, and it's kind of like a web, a gooey web surrounding the cell. So plants, fungi, and bacteria have cell walls to maintain their shape and support. So they have kind of like a hard wall, and then they have the plasma membrane inside. And this is a ECM of types, a cell wall. But animal cells don't have those. So in plant cells and fungi and bacteria, they have a primary or really thick cell wall, kind of a middle lamella, which is like the glue that holds their big, heavy outer cell wall, and their secondary cell wall, their thinner cell wall that's bound to the plasma membrane. Animal cells, on the other hand, have this gooey matrix made of different chemicals. They have long fibers of collagen, which branch out like this to provide a buffer or a surrounding, and they have things called glycoproteins, glyco, sugar, proteins, protein, which are proteins stuck onto these fibers, these collagen fibers, with long chains of carbohydrates or long chains of sugars that are sticking out of them. And they act as means for animal cells to stick together, or for animal cells to recognize each other. In addition, animal cells have these proteins squished into their cell membrane called integral proteins or integrins. And they're involved in signal recognition, which we'll talk about in the next unit, and also sensing around in the environment and responding to stimuli. So this network of collagen fibers and glycoproteins and integrins all act as ways for the cell to sense what's out there and respond to it, and also to hold on to its surrounding environment. All right. Lastly, for this unit, there are the junctions between cells. So cells in your body are all stacked up next to each other, and they're joined next to each other in three ways. One, they can be joined, like in this picture, in what's called tight junctions. Their plasma membranes are literally stuck together, right squeezed up next to them, with only these small little tight junction proteins helping to bind them together, but pretty much they're plastered up against each other, like if you stuck your face up against a window. In other cells, we have what's called desmosomes, which are protein or keratin like rivets or anchors that like bolt two cells together, kind of like this, that bolt the two cells together and connect that junction, that keratin junction, to the cytoskeleton or the backbone of the cell. And third, other cells have gap junctions, or little protein pores or channels that directly connect two neighbor animal cells together. And it's very important for communication and sending chemical messages back and forth. Now, a cell might have all three of these different types of junctions, or it might just have one. In plant cells, what basically is gap junctions are holes in the cell wall in plants are called plasmodesmata. And these things we'll talk about a lot more when we talk about how cells interact with each other and how they maintain homeostasis and how they signal. Alright, so that was a review. A lot of this may be written in bio. I know the junctions and things might be new, but not too deep in content, just something to review, and we'll look at specifically the cell size in our lab on Friday. If you have any other questions before Wednesday when we talk about this, just email me, let me know, and I'll be sure to help you out. I hope that was helpful.